The overall goal of this experimental approach is to simulate a ferruginous upwelling system and to detect geochemical profiles of oxygen and aqueous iron 2 in the water column when cyanobacteria were present and produced oxygen by photosynthesis.
This newly developed method can help answer key questions in the field of Earth's history, such as the oxygenation of the atmosphere and the deposition of banded iron formations. The main advantage of this technique is that geochemical conditions, as they might have existed in a Precambrian ocean, can be simulated on lab scale, being investigated in situ with modern methods.
Though this method can provide insight into early Earth studies, it can also be applied to other modern environments, such as for geomicrobial investigations and chemical fluxes in sediment bodies. Generally, individuals new to this method will struggle because setting up the experiment is not trivial, but the video will provide information on the most critical steps performed in the protocol.
Bacteria culturing, early assembly steps of the setup, and notes on sterilizing materials are all described in the text protocol. Note that the cell solution for Synechococcus 7002 must be anoxic for later experiments. Therefore, remove the soluble oxygen by bubbling the solution with anoxic nitrogen carbon dioxide via a 0.22 micron filtered syringe, having a second needle in the stopper to release overpressure. In order to stop photosynthesis and the production of oxygen, cover the culture bottle. After 5 minutes of bubbling, flush the headspace.